actual motif region is less important. So probably the role of cofactors becomes predominant in the case of macrophages. In ES cells, these regions surrounding the motif are roughly continuous, and same is true for cancer cells. And we already know that ES cells and cancer cells should behave more similarly to each other, and the two immune-associated cell types should behave more similar to each other, and that is what also emerged from the analysis of the DNA shape of these sequences surrounding the transcription factor binding motifs. So then going from stat 3, we went to look at the large number of transcription factors, 1,500 transcription factors, and their ChIP-seq data. Their NGS data analysis showed that there are 4 clusters, and they have different types of profiles in terms of the information distribution around the binding motifs. And so there is a group of 229 transcription factors which have a sharp peak where the surrounding regions are not important. Then there are 504 on the other extreme, where the surrounding regions play the predominant role in the interaction with the protein, in the protein DNA interaction of transcription factor. There is another resource that comes from our analysis of the machine learning application to DNA binding proteins, where we try to integrate the gene expression with the sequence information to predict DNA binding proteins. And this became possible. So just I show you this data, that there are the genes that code for DNA binding proteins have typically higher expression value, but this higher expression value is not necessarily be associated with DNA binding activity. This is in combination of these gene ontology terms, we could integrate the gene ontology information about the co-expressed genes and the nature of the co-expression, and we could improve the annotations of DNA binding proteins. Actually, the integration of the expression data and the sequence and the structure data helps us to annotate proteins in the specific context of the expression profiles. That is, in the classical annotations of proteins is like if this is the sequence and this is the structure, this is the function of the protein. But when this function will actually take place, under which cellular context will this function will be performed, has to be modeled with the help by taking cognizance of the expression signature or the cellular context. So cellular context can be incorporated into the model by the expression profiles. So we try to combine these two and predict the DNA binding function activities with the help of these two features taken together. And we showed that there are some proteins which do not have a very strong sequence level or structure level signature of DNA binding. But if certain genes are co-expressed in a given cell, they may perform this function. So this kind of a mechanism that the functional compensation which is encoded in the sequence, if the functional encoding in the sequence is not strong enough, that can be compensated by the expression signature of the cell. And this will allow the different types of transcription factors or the proteins to perform different functions in different cell, because the other expression environment also contributes to this same function. Then we applied just a few words about deep learning models. The models that I showed you so far, most of them were the classical machine learning models. In the deep learning model, I have a few words more to say, just to put it in a background, that we have so much of data everywhere, and then we do not have, I just give you the example of dimensionality. Again, I was saying that we are collecting a lot of features for the data, and we often talk about big data, but sometimes we do not have big data. So we have a lot of data sometimes, and we often say in biology there is no data for this. So actually, that there is data required is not what is available. So we have to use the data that we have, and we try to predict and try to model information that is of our interest. So people have been collecting indiscriminate features. For the kind of models that we have to create are such as in MRI images. Even if it is a 600 by 300 pixel image, you have 1,80,000 pixels. This is the dimensionality of my predictive model that I'm trying to create. If I do an ECG diagnostics, these are the problems that we are addressing in our labs. That's why I picked up these problems. So if we are doing a time series analysis of an ECG, and I want to know where this ECG has become erroneous, where is the location of erythemia, then I have again a very high dimensional. If I have a 1 hour recording of for every second, which is the number is, as you can see, 3600 per hour. And then the thing is, this dimensionality itself may not be a problem, but if we are trying to fit models, and if we have just 10 instances of data that we want to predict to model, and in this case of MRI images also, we do not have more than 100 images. Sometimes we have just 100 images. Like if we want to predict, like we were working on one problem where we wanted to relate the MRI lesions observed in MRI to the linguistic deficiencies. So we had less than 100 images. So the challenge here is same is true for the gene expression data analysis. In all these cases, if you look at co-expression, you have millions of dimensions and just few hundreds of the data instances. So if you have just a few, the number of data instances is fewer than the number of dimensions. The conservative, the classical approach is that the number of data points that you want to fit your model should be more than the dimensionality. But then you see so many models coming for image recognition, image classification, and all this. If you are feeding the entire image to the model, you are really feeding a lot of features into the model. And then you are trying to, you do not have enough data to train in the classical context. But then how these models become possible? These models become possible because of something that is called convolution. I don't know if I have that slide here. Yeah, so the convolution will try to first, instead of directly putting into the model, the convolution allows to create a structure within these features. So in some sense, the inherent power of the CNN comes from the fact that the features can be convoluted, and they can actually, the data requirement becomes much less.
You
If
have
we
5
were to
minutes.
fit 5 minutes, okay, yeah,
Okay.
okay, thank you. So this the primary challenge for creating these machine learning models or any high dimensionality models is to avoid overfitting, so that generalization can be achieved. So this generalization can be achieved with the help of CNN. Have been particularly successful in this field because they have an inherent quality of modeling the relationships between these features without actually directly training them. So the convolution layer makes this possible, and then later on they are pooled so that the effective dimensionality can be much less than what it actually, the number of parameters we are training. So this is where we studied the comparison between the shallow, the traditional, and deep learning models for cognitive performances of stroke patients. And that is where we systematically showed what is the data requirement. If we create our model with 50 MRI images, 60 images, 80 images, how the performance goes. And what at the smaller data sizes, classical model work were better. But there is a certain data amount where the deep learning overtakes, and that number can be estimated by a systematic analysis of the modeling the models. And then we used this combination of principal component analysis and the feature based, the CNN, and that this is what I'm showing you, what I was talking about, the convolutional layer, which creates these smaller units, and then it creates a new representation of features. And finally, we use a fully connected layer, which has a much fewer units than we had in the beginning. So we have in the input model 61 into 73 into 61 3D image of MRI, and in the end, actually fully connected model has just 500 units. So this entire modeling from the convolution to the pooling and the fully connected layer is where the actual interaction between pixels is being modeled. And that is what gives power to CNN in contrast to a traditional deep model of multi-layer perceptron. So I skipped these results. So this is what we do. We did the comparison. So this is another problem we dealt with the evolutionary profiles of proteins, and we try to predict again their protein functions. And this deep model with the help of CNN performs much better than all the previous models that we have also developed earlier and other researchers have developed. This is ECG I already talked to you about, and here we use the model, the deep model, which is an advanced version of the recurrent neural networks, LSTM, and this combined with the classical models of SVM could predict where exactly the anomaly in an ECG is and what kind of anomaly is in a given ECG signal. So in summary, I summarize this, what I said, data science has always been fundamental to the scientific analysis and discovery. the field has moved from simple statistical modeling to classical machine learning, and now the deep learning models, and also the natural language processing, which I have not touched upon today. So the technologies that are used to this have changed not only in terms of the mathematical and computational model, but also in terms of the infrastructure is created. So now instead of doing this on the mainframes computers, people were doing on mainframe computers earlier, then moved to servers, and now they're moving to clouds. So these from clusters to the public clouds. So this is how the paradigm shift has not just taken place in the form of computational models, but also in terms of the resource planning and the infrastructure that is used for computing. And a brief discussion I had was about the shallow and deep models. And it is not always that the deep learning model will perform better than the shallow or the traditional classical. It depends on the model complexity that we want to generate, that the data that we have, really it has that kind of a complexity, and how much data we have. So these have to be taken care of, and we cannot blindly believe that a deep learning model will always do better than shallow or classical machine learning or even simple statistics. That's the conclusion from today's discussion. And finally, I conclude with this little poetic description of this: mimicking brain, shallow and deep, machines learn when man is asleep. Without complex equations or laws, making use of errors and flaws, that is how the training takes place. Regressive, divisive, and classifying, these are the terms often observed in machine learning. So regressive, divisive, and classifying only in AI are so gratifying. So this is the summary. And finally, people who have contributed to this work, some of my lab students, some of them may not be represented here, some of them have left, a couple of them have left the lab since, and but I acknowledge them because their work is included here. So thank you. That I stop here.
Okay, thank you very much, Professor Ahmad, and then very interesting and enlightenment talk from your group. And I noticed there are some questions on your talk. Let's we continue to the Q&A. Yeah. And I think one question and directly you can answer directly. Okay. Okay. The first question from Umi Baroro from Indonesian School of Pharmacy. Yeah. He would like to ask about the modeling of DNA or RNA. So the question is, how to evaluate our model of DNA or RNA is good. So is there any parameter that can that we can use to scoring our model?
So is this the question, Umi Baro? So I can read it directly from the screen also?
Yeah, yeah. Umi Baro, yeah.
So hello,
would like to ask about how to evaluate.
I would like to ask about modeling of DNA RNA. So in general, I'll say give a general answer so it can help other researchers also. How we so the question is how we evaluate whether my model is working or not, right?
Yeah.
So there are this is a fundamental question of machine learning, because my model may be working very well on the data I am training. That is not what we want. We don't want our models to memorize. We want our models to learn. And there is a difference between memorizing